So thanks, Mike. My lab at the Technion, I've been there for 2.5 years, and my lab works on different lipid-based drug delivery systems, and some of them have already actually been translated to the clinic. Some are translated to agriculture, and others are more basic, and we'll start with a basic example and maybe a specific application of one of these systems. It's a system that can actually produce proteins on site. And what we envisioned in our mind, many times we use drug delivery systems or nanoparticles, nanotechnology, to take an active molecule from one region to another and target a specific area. We asked ourselves, could nanotechnology also act as small bioreactors, small systems that would travel throughout the body, and only where a drug is actually necessary, we'd produce it on site. So we'd have a small factory that can produce a drug once triggered to turn on. And the system we decided to develop is a lipid-based system. So we have a liposome, and it can be of different sizes. And what we have inside are all the molecular machines that are necessary for decoding DNA and actually producing a protein on site. And what makes it useful is actually being able to turn on the system only where it's necessary. So we'd have the protein produced only where it's necessary. And the system up to here is actually an inert system to the body. The body recognizes these molecular machines, which can be amino acids, ribosomes, RNA polymerase, etc., which exist in our natural cells. However, the protein is what makes it potent, and turning on the particle on site is what changes it from just a bioreactor to an active bioreactor. So what we envisioned in our mind is a system that's injected intravenously. The particles actually distribute throughout the body, and where necessary, we'd trigger them to produce the drug of interest. And in the other regions of the body, these particles would actually be degraded. So our first proof of concept was of particles in the micron range, and you can see here approximately a 5 micron particle with the production of green fluorescent protein inside of it. You can notice the size of the particle and the protein actually being expressed inside the system. And we asked ourselves, how small could such a system be and still be capable of producing a protein on site? And what we found is we could go down to around 170 nanometers, but once we went any smaller than that, the particles became inactive, meaning they didn't have all the components that were necessary for producing a protein inside of the system. And when we looked a bit closer at these particles, what we found, it's not the molecular machines, it's not the ribosomes or the RNA polymerase. It's actually it's the DNA that's hard to fold into these particles. For example, this is DNA. It's very narrow, but lengthwise it's rather long, and it has to assemble spontaneously in a manner that would enter the particles. The ribosomes, you can see here, this dark area within a particle, are around 6 to 20 nanometers in diameter, so they don't have any problem entering these particles. But the particles we used didn't have histones or any active moieties for condensing DNA. That's why that was the size limit we had there. So that doesn't give us the specificity. And what we were lacking was the switch to turn on the particle and actually be able to produce a protein on site. And what we added is a cage molecule that sits on the DNA. The cage molecule is sensitive to light at specific wavelength, and only when light hits the cage is released, and then the DNA can start being decoded into RNA, which will then be decoded into a protein. And you can see here that we get robust protein production after releasing the cage. However, the cage isn't perfect, and we have some leakiness in the system, and that's a default that we have that's still embedded, and it seems that this is as good as we can actually get. And as an engineer, one nice thing we were able to do was to turn on the particles, then we can stop turning them on, and then we can turn them on again to produce protein over time, only when they're actually necessary in site. So this is what it looks like when we turn on protein production inside an animal. This is the production of luciferase inside of the particles. On the left-hand side, you can see a mouse that was irradiated with light and the site of irradiation. On the right-hand side, also a mouse that was injected with the particles. You can see that some protein is being produced. However, it's not as robust as the animal that is exposed to light, and the protein is produced on site wherever it is necessary. So why would we even want to go in this direction? And what it gives us a lot of modularity as a system. We can tune the particle size. We can add targeting moieties to the surface of the particle. And we can also choose what building blocks we encode inside such a particle. For example, we can go only halfway. Instead of producing a protein, we can produce RNA. In this case, we produce a short hairpin RNA, and we introduced the particles to cells, and then we were able to knock down a gene inside of a cell. We can use it for producing a very toxic protein, which we wouldn't want to have anywhere else in the body, and then actually reduce or kill cells only where they're necessary. In this case, triple-negative breast cancer cells. But we could also envision another approach in which a nanoparticle can actually perform a therapeutic task inside the body. And what we envision in our mind is replacing the surgeon's scalpel with a nanoparticle that actually sees diseased tissue and can cut it with molecular precision. And the medical site we chose to go for was actually orthodontics, where many of us have undergone this long treatment, and we thought we could use nanotechnology to reduce the time it takes to align teeth, specifically reduce it from 2 years to 6 months. And the biological space we were working at, it looks like this. So we have the tooth that's in the mouth and the underlying bone. And actually what connects the tooth to the underlying bone are collagen fibers that keep each tooth in its current location. When people undergo orthodontics, these fibers, these collagen fibers, have to be stretched to their new orientation. And we asked ourselves, could we use a particle that would express collagenase and that would degrade the collagen fibers we have inside the oral cavity? And that way we'd enable the tooth to move more easily or more freely to its new location using orthodontic treatment. And what we developed initially, these are particles that contain collagenase inside of them. We're not producing the collagenase yet. And you can see here it's a nanoparticle. It has collagenase inside of it. And in order to interface with the dentist or the orthodontics in a simple manner, we embedded these particles inside of a chip. And the chip is placed in the sulcus, which are the small pockets we have near our teeth. And then in an illustrative manner, what happens, the particles or the chip is inserted, the particles are released, the collagenase out of them, and the collagen fibers here are actually relaxed, and the tooth can move to its new destination.
Does it fall out?
No, it doesn't fall out. Yeah, if you put a lot, you could maybe do that. Yeah, but we tuned that. So first, we looked at the effect of collagenase on collagen fibers. On the left-hand side of the screen, you can see a collagen fiber before being treated with collagenase, and on the right-hand side, after being treated with the collagenase nanoparticles. And you can notice that the collagen fibers are actually relaxed. And when we came to align teeth, so we do orthodontics to rats today, and what we did is we inserted the regular spring that's used in human orthodontics, and we tested the ability of the rear teeth, the molars, to move towards the front incisions. And this is what we measured over time. You can see it here both in X-ray and in the rat's mouth. And to make a long story short, on the right-hand side, you can see after 15 days of treatment, these are regular orthodontics, and these are the enhanced orthodontics we have here. So after treating with collagenase, you can actually enhance the tooth mobility to its final destination. But that wasn't the end of the story, and maybe answering the question, then what happens after you reach that site? And what we found is that the collagen, if we started here and then we relaxed it, it actually healed very well after being treated with a biological tool, because we're using the same tools the body actually uses for relaxing the collagen fibers. So when we looked at the profile of strength of these collagen fibers, they actually returned almost to their initial state. And this looks a bit, to me at least, like a broken bone after healing at the final destination. So what we would have is not only faster motion of the tooth to its final destination, we'd have less relapse of the tooth to its initial destination, meaning people wouldn't have to wear the 2-year follow-up after the treatment. And biologically, what we saw is that we're looking now at 4 fibroblasts that are on a collagen fiber. So these are the fibroblasts. The collagen fiber is here. We can't see it's outside the focal plane. And this is before exposing the collagen fiber to collagenase. And now you'll be able to see what happens to these cells over time as we expose the fiber to collagenase. and the cells change their orientation. So if we started with a stretched collagen fiber, now we have a collagen fiber that's relaxed, but the cells don't fall off. And what happens now is that all the repair mechanisms that these cells have, the fibroblasts have for repairing collagen, kick into action. And these are 3 examples of genes that start working immediately to repair the collagen at its new final destination. So we embed I'd say the treatment, but we also embed the healing once it ends. So we looked at the histology, and the histology was very similar to regular orthodontics. We get some mild inflammation, which is expected and is actually necessary for the healing process on site. And we saw that not only the material healed, also the hard material. So this is an untreated tooth. This is a CT of the rat's head. And on the left-hand side, you'll be able to see, this is the arc you see here is the remodeling of the bone actually at the new orientation. So this is necessary for the tooth. The bone, underlying bone, has to rebuild in its new site. So we got healing both of the soft tissue, but also of the hard underlying tissue, and that pretty much makes the treatment effective. So that was one story we have from our lab. I want to thank you very much for listening. This is my team. Many of them are undergraduate, but some are undergraduate, some are graduate students that participate in this work. Asaf and Nadira, actually the leaders in this study. Thank you very much.
Well, I'm frankly amazed, you know. Could I ask about the collagen structure? It's slightly inferior. How does it compare to the normal process, so to speak?
Of the collagen or the collagenase?
The collagen.
Okay, so we take it in an oral cavity, or
well, you showed pictures there, and it had gone down, and then it came almost
yes,
back up.
Right.
If you used the more traditional, slower process, would it look the same or back to the normal?
So actually what happens, and that's one of the major issues people face today in orthodontics, that the teeth have a relapse and they return to their original location. Meaning the collagen doesn't rebuild in its new location, it's stretched, and then it wants to pull the tooth back to where it used to be. And people have to put in a retainer at night for several years to keep the tooth in its new site. Using this, we actually don't need to give that treatment, and what happens is we degrade it on one side or relax it, and then rebuilds in the final orientation, the orientation we want to have. So
it's impressive.
Yeah.
Fantastic talk. I did, thank you. Fantastic talk. Question for you about the that first set of data where you essentially make your own proteins in a liposome, which I find absolutely fascinating. What do you think about the pros and cons of delivering the entire machinery or delivering mRNA, say, for example, to an existing cell? I mean, clearly what you're doing would be multi-steps more sophisticated than the alternative, which is just the mRNA delivery. But maybe you can just give me the pros and cons of both approaches.
Yeah, so thanks very much. I think there are pros and cons to each one. For example, delivering mRNA, as long as you keep it intact, you know that, and it reached the cell machinery, and in a good form, you'd actually be able to have the protein produced. However, it would be very hard to know how much of the protein would actually be produced on site. So for certain medical applications, we want to know exactly how much of the therapeutic compound we have on site, and we don't want to have production just going on. So I think that's one difference we'd have between mRNA and protein delivery. I think both applications, though, or both approaches are good. And I would say if we ask ourselves from a clinical perspective, most likely that delivering mRNA will reach the clinic before such an advanced system, or maybe sophisticated nanoparticle would probably ever see a patient. However, there are certain things we can learn about protein production and about how different cellular machines work in a confined area and maybe inside the body using this nanoparticle approach. If you ask me, what would, if I'd be an investor, VC today, what would I invest in to bring tomorrow to the clinic? Most likely mRNA. But if you ask me maybe 15 years from now, I'm sure we all want small robots inside the body that can actually perform a therapeutic task in a very intellectual manner. So I would say this is for tomorrow or 2 years from now, and one is for maybe 10 or 15 years from now.
Well, there's the regulatory and immunology issues as well, I guess, as to whether this is a host protein or not a host protein.
Absolutely, and then we can use machines though from the body itself, from the patient, right? And that's one approach we're looking at, yeah.
Any more questions? One more back there.
Hi, great talk. Quick question. Did you guys get to use your chip in disease tissue? So tissue where the collagen fibers were lost already, such as periodontal disease?
No, yeah, we always treated a healthy rat and or a healthy collagen fiber. And the question, would it actually induce a repair of something that may be diseased? A huge question that we very much want to answer, yeah.
Great talk.
Thank you.
Okay, so we move now to Stephanie.
Let's put this on first.
We'll have time at the end for questions. We're doing extremely well.
I don't know how this works.